pretty amazing talks so far this morning. You know, we really have this sort of contextualization here, starting off with, you know, what do we need to do from a scientific perspective of rare tumors? How do we need to design better clinical trials? And now this inspiring talk from Kim Rathmel on where are we in the state of our being as scientists. And the future really is bright, provided we are flexible. I really wanted to call out, you know, kidney, in terms of where we are in this room, you know, this group has grown and grown. Why has it grown? KidneyCAN has gone to the House, has talked to individuals, has motivated the funding through CDMRP, the KCRP, where we had, up until very recently, $50 million in funding, which has resulted in these mostly young, brilliant people working on somewhat of an orphan disease. And the progress that's been made in the past 10 years because of this funding has already been astonishing. And we know that the multiplicative effects of this as we move forward are going to be even greater. So hopefully we can get through, we will persevere, and we will continue to grow, and I think it's going to be amazing. To that point, what we're going to be talking about today are a couple of examples of funding that has been granted to various individuals and organizations. And we're going to start off by having my session. And the way this works, this is going to be very quick. Okay, we're going to have 5-minute talks, and we're going to have 5 to more minutes to be able to actually Q&A each of the speakers here. Okay, so be on your toes, have your questions ready. We have an hour, and at the very end, we're also going to sort of have a final sort of discussion. So without any further ado, basically, first of all, wanted to introduce my co-moderator, Dr. Cheryl Walker, who's at Baylor College of Medicine, who has been a friend and a colleague for the past decade or more, and has really been a mentor to me and to many other people in science. Let's start with my presentation. So what I'm going to talk about here is one of the CDMRP funded grants, which is the Kidney Cancer Research Consortium. So this really was the brainchild of a number of the physicians in the room, saying that what we really need is we really need an infrastructure to allow us to be able to work collaboratively across institutions in the US and hopefully perhaps even broader, to be able to get people to work together to answer the tough questions that perhaps are difficult through industry, but can be facilitated through industry. And the team that we've developed is the one that you see here. We have investigators from MD Anderson, from Penn, from UT Southwestern, from Duke, Vanderbilt, Michigan, and Beth Israel. And beyond this, we also have a number of individuals who are more junior, who are collaborators. And so the group of individuals that are involved in this is actually quite large. Our mission really was to develop hypothesis testing and generating clinical trials that link identifiable biological characteristics of renal cell carcinoma to specific treatments and treatment strategies. So what we really wanted to do is develop tissue-rich, information-rich, biomarker-rich trials that, although they may not be 1,000 patients large, will have sufficiently large data sets that will arise out of them that they're going to be larger than the sum of their parts. And what we really needed is an infrastructure to do this. And the infrastructure is what we've really been working on over the past few years to develop and perfect. We have an organizational structure that you can see here. We have a steering committee on the left of the investigators that you saw, as well as 2 patient advocates, one of whom is in the room today, Brenda Knapp. We are overseen by the CDMRP. You can see in the top sort of right there. We have an EAB membership that I'm going to be talking about before, and then we sort of have our quote-unquote clients, which are the clinical trial sites. We have an amazing advisory board. we've got Michael Atkins, we've got David Bottaro, Sergio Garza, Brian Lewis, and Nazim Zazwala. And these are individuals from various areas, either academia, informatics, as well as pharma, to really make sure that we stay on the straight and narrow. And we have a portfolio of trials that has grown, again, through the support of industry to quite a large number of studies here. And we have studies that have already completed accrual. Katie Beckerman, who's in the room, is presenting this as an oral abstract at ESMO for her study. And we have a spectrum of studies here that look at immune modulating agents, look at theranostics. We're looking at specific disease states. And you can see that what we're also doing is we're trying to make sure that these are biomarker rich, and also developing patient-reported outcomes measures that are going to be integrated in each of these trials. So the sum really should be greater than the parts. We're looking at a variety of, we're looking at this biologically in terms of the different areas of the tumor microenvironment and interacting with this in different ways. Because of the strength of the institutions, we have research hubs that are sort of developing across these various institutions that allow the institution-specific expertise then to be brought into our particular into our particular coordinating center and the overall effort. And we have domains of interest that are being led by various individuals as well. Critical things like biomarker development, junior faculty development, underserved populations, rare tumors are being led by each of these various individuals in the group. We have close relationships with philanthropic and foundational groups that you can see here. And we've really made progress. As we see, we have these 10 studies that are in various stages of development. We've completed some, a number of them are opening. We have relationships that are developing from an informatics perspective. There's an enormous amount of information on renal cell carcinoma patients that exist within each of these institutions that we really need to be able to take advantage of beyond the clinical trials. We need to be able to develop therapies and also biomarkers in the next decade that are going to transform renal cell carcinoma research. So this is my last slide. We have a wonderful website that's been developed by some really talented individuals. I invite you to look at it. So I'll stop there, and thank you very much for listening. Question?
Is there a possibility of sharing tissues or materials through this consortium? Sorry.
So the question is whether or not there's a possibility of sharing tissue. So we have the, what I call a federated tissue collection, that where the tissue that's being collected for these different clinical trials sort of exists within each institution. Then when the investigator needs to perform analyses for their particular study, that will then be brought to that particular investigator. But part of what we really want to do is we want to expand that federated tissue collection and blood collection to other investigators that are outside of the consortium, where we'd want to be able to allow specific hypotheses to be brought forward and that could potentially be tested through the resources that we have within the institution. So happy to talk about that further.
Eric, I have a question. Tell us how the defunding of the KCRP program is going to be affecting your consortium.
Right. So because of the funding cut for this FY, we actually had to scramble quite a bit to be able to get funding for the next the remainder of our 4-year period. We have actually been successful in securing funding for all of the clinical trial sites through 2028. But we're missing 1 year of the funding for the actual coordinating center itself. And so we are going to have to work really hard on making sure that that is actually going to be secured in the end. So we are somewhat compromised, but right now are in reasonable shape. Okay, there are no further questions. Let's move on then to the next talk that's going to be by David Braun.
much to the organizers for the opportunity to speak today. Excited to be back here and see some familiar faces. Really a wonderful meeting. So what I'd like to talk to you about today is a little bit about a personalized cancer vaccine for renal cell carcinoma that really was supported and developed through the Kidney Cancer Research Program. And so I'm going to talk a lot about the science, but also really want to highlight the support that really made that possible. And so to frame this talk, just my sort of simplified worldview of anti-tumor immunity in kidney cancer is this idea of driving CD8 T cells towards the tumor, and how can you get them there as quickly and as accurately as possible. So traditionally, that's been through immune activation methods, either immune checkpoint inhibitors to take your foot off the brakes, or by pressing on the gas pedals, historically cytokines, and now lots of immune agonists. But the other side of this is really the immune navigation component, which is how can we not just turn on the immune system, but really steer it in the right direction? And that's really, in my mind, through antigen-directed therapies. And I think personalized cancer vaccines are really an ideal example of how to do that. And so why did we sort of start off exploring this study? Well, we know that neoantigens are effective targets of anti-tumor immunity across many different types of tumors. We know that neoantigens have elicited T cell responses in things like melanoma and even in pancreatic cancer or glioblastoma. But kidney cancer represents a bit of a unique challenge. While it's both historically and contemporarily immunogenic, at the same time it has a very modest mutation burden, so not a lot of neoantigens to target. And we know that there's really a challenge in improving the outcomes for our patients in the adjuvant setting. Adjuvant pembrolizumab has really been a major development, but there's an enormous amount of room to go to improve outcomes for our patients. And so this is what really motivated our study to ask, can we design an immunologically effective, personalized cancer vaccine targeting neoantigens in kidney cancer? And so the study design is a small study. This was 9 patients with high risk but resectable disease, so either stage 3 or stage 4, clear cell renal cell carcinoma, but all of the disease being removed. We sequenced the tumors, we predicted neoantigens, and we created a vaccine consisting of synthetic long peptides containing up to 20 of those neoantigens, divided into 4 pools. And we gave it together with immune adjuvants in a sort of typical prime boost format. So similar to how you think of infectious diseases, a prime to really activate and prime T cells, and then a boost to try to build long-lasting memories. So the questions we had were pretty fundamental. The first one is just, is this feasible? Can we actually do this for a low mutation burden tumor like kidney cancer? And the answer is yes, that despite a modest number of coding mutations that we can see at the top, for every single one of these patients, we could create a multi-epitope vaccine that targets not just single nucleotide variants, but frame shift insertion deletions that could target in the large majority of patients driver mutations in kidney cancer. So now that we know we can do it, the question is it actually immunologically effective? Are we actually steering the immune system? So this heat map summarizes a lot of data, interferon gamma LE spots for peripheral T cells. And the basic question was, are the T cells actually reacting to these neoantigens and capable of recognition? So there's a lot of data summarized, but if we look at all the way on the left-hand side, that column 0, that's all of the patients before vaccination, and we see it's almost entirely white before vaccination. So there's no pre-existing, at least detectable levels of immunity against neoantigens. But for every patient, as we move from left to right, we see that red come up, indicating that we're generating a neoantigen-specific response in the peripheral T cells. And we can have one example of that here. This is patient 101, where we see that for 3 of the 4 vaccine peptide pools that we administered, while there's no pre-existing immunity, there's really strong peripheral T cell responses through the vaccination period. And we can go even further and ask which are the specific neoantigens that led to those responses and deconvolute those up to 20 different targets. And that's what we've done here. And perhaps a little bit to our surprise, what we observed was it wasn't just random mutations that seemed to elicit effective immune responses. In many cases, it was the kidney cancer driver mutations that we know so well. In this case, PIK3CA and VHL. So we give an example of one of those interferon gamma LE spots. We can see that here, where each of those dots represents one reactive T cell. So we see the negative controls, DMSO and HIV peptides, essentially no dots. We see our positive control, PHA, so lots of T cell activation. And our reaction against the VHL mutation, our vaccination, shows tremendously strong T-cell responses. And it wasn't just VHL. If we go across common kidney cancer driver mutations, PVR1, PIK3CA, KIDM5C, we see strong responses against the driver mutations. So yes, it does elicit an effective immune response. Now, is it durable? Is it long-lasting? So to answer this question, we tracked vaccine-specific T-cell clones within the peripheral blood, and that's the red line here. So at the beginning, we see they're basically undetectable, at the lower limit of detection. They rapidly rise up with vaccination, and through the boosting phase, we see those persist. But I think what's fairly remarkable is even after we finish the vaccination, so months or even years later for some patients, we still see persistent T-cell clones that are vaccine reactive. So it is really listing a durable T-cell immunity. And then critically, is it actually capable of anti-tumor activity? Is it capable, are these T cells actually now capable of recognizing the tumor? So we took those same vaccine-expanded T cells and actually put them back on top, in vitro, on top of those patients' own tumors. And we again measured were they reacting. And it wasn't 100%, but in 7 of 9 patients, we detected vaccine-specific T cells that were capable of reacting against autologous tumor. Now, is this clinically efficacious? We obviously can't answer that with a 9-person study. And so this is huge caveats. We obviously are really interested. That's the thing that ultimately matters. I will say we're encouraged by the fact that all 9 patients, despite having high-risk disease, remain free of kidney cancer throughout the study. 1 patient unfortunately died of a completely unrelated cause, but no patients had a recurrence of their kidney cancer. So this is certainly encouraging, but it's only 9 patients, and so we really need to follow this up. And so to summarize, these neoantigen vaccines are feasible for kidney cancer. They can elicit effective T-cell responses and anti-tumor immunity. And I think at least this preliminary signal of clinical activity really sets up INTERPATH 004, the next study that will really hopefully demonstrate some clinical activity. With that, lots of people to thank, lots of collaborators on this project, Kathy, Patrick, Darren, Tony, the folks in my lab, many of whom are here today presenting posters tonight, my collaborators at Yale, the patients and their family, and then really particular sort of call out to our funders, the National Cancer Institute, but really the Kidney Cancer Research Program. Both myself as a beneficiary of the Academy of Kidney Cancer Investigators, led by Braun, has been a tremendous resource for me to be able to develop as a young scientist and as a physician. And then this work, particularly the scientific aspects of this work, was all enabled by funding from the Kidney Cancer Research Program. So I remain very grateful, and hopefully this work will continue. Thank you so much.
Questions from the audience?
Anuzar.
Great presentation as usual. The melanoma folks have been ahead of us in RCC in this field, as well as other areas when it comes to immune therapies. What has been the lesson learned there from melanoma? Have they, that you can apply to our field with their vaccine strategies, neoantigen identification, and
It's a great question, and I think we've been really the beneficiaries of a lot of that experience within melanoma. So within neoantigen vaccines specifically, this, as you said, all began with melanoma, both from the BioNTech group and from Kathy Wu's group in 2017. And so for this study, we actually did learn a lot of those lessons in terms of how to predict the effective targets, what are better ways to predict antigens, what are potentially better ways to administer the vaccine as well. Melanoma is further ahead. They've completed their phase 2 study, which was a successful study led by the late Jeff Weber, and is now already in a phase 3. But I think kidney cancer is not far behind. I think the idea of sort of learning those lessons, we have to recognize kidney cancer is obviously different. We have an order of magnitude fewer mutations or neoantigens, but there's still valuable lessons to learn.
David.
Hey, Abhishek.
Hey. So a couple of questions. One is a clarification of what the ABCD peptide pools mean. Are they the same pools targeting the same repertoire of neoantigens, or different subsets that were designed over here? And then related to that, given the fact that the original training was developed on the grade 3, grade 4 tumors, now granted that even there the mutagenic profiles are probably lower than many other grade 3, grade 4 cancer types, but still would be much more than early stage cancer. If you can comment on how this strategy would be relevant for a person coming in with a more manageable early grade tumor, if you will?
So I think both great questions. So the first was, what were those vaccine pools? So it's basically, we picked up to 20 different targets or neoantigens, and then we distributed them into different pools. So the same 5 were in pool A, the same 5 were in, a different 5 were in pool B, pool C, pool D. The reason we did that was a concern of one epitope being immunodominant, that we thought that that might come from essentially competition at the local lymph nodes. So if we had one target that was really dominant and we put all of them together, it would sort of outcompete everything else. So by spreading it across different draining lymph nodes, we thought we could sort of address that issue of immunodominance. The second question is how well this might translate to earlier stages of the disease. It's a fair question because we know the immune composition is very different than those early stage tumors. And so I don't think we'll know till we try. I think at the same time, some of those earlier stage tumors tend to have really excellent outcomes. So I think it's not that this would be for every early stage tumor, but if we can really identify ones that are maybe higher risk, those would be the ones to sort of think about how to target.
One last question before we move on.
Ari.
Yes, it is a great presentation, David. Just a question on your thoughts on the critical role that is going forward in the right and the right clinical scenario.
Yeah, it's a great question because there's certainly a long history of not neoantigen vaccines, but cancer vaccines in general, and unfortunately a reasonable history of lack of success. I think part of it is the target, so neoantigens really represent a different target, but part of it is the disease setting. And I think even for neoantigens, as we see cases where it's been tried in more bulky metastatic disease, we haven't seen sort of convincing evidence of success. But if we look in kidney and pancreas and melanoma, those signs of success are really in that minimal disease setting, that minimal residual disease setting, such as the adjuvant setting after surgery. And so I think it's maybe, at this point, maybe too much of a lift for vaccines, certainly by themselves, to do something the metastatic setting, the grossly metastatic setting. There you have to think about co-therapies that might be effective. But I think the vaccines have a real chance in that sort of adjuvant space, and broadly in this sort of minimal disease state. I know Chad and I have talked a lot about, in oligometastatic, these sorts of situations where there's minimal and manageable disease, but high risks of recurrence. I think that's kind of the sweet spot for these vaccine approaches.
Great. Thank you so much. All right, next we get Dr. May Ko up to the podium. She's at the University of Utah, and she's also the CEO of CUDA Therapeutics, and she's going to talk about some of the work that she's done.
Thanks for the introduction, and I'm privileged to be here. I'm glad to share with you our story of discovery in developing, so we can advance this, novel ferroptosis inducers for kidney cancer. And this is work done actually at my company, CUDA Therapeutics, but supported by basic research at my lab. And so I have some disclosures on the slide here. So many of you may have heard of ferroptosis. For the uninitiated, it's a form of regulated cell death driven by iron-dependent lipid oxidation. And what makes ferroptosis important or exciting is the fact that once initiated, it goes through a chemical chain reaction. So there's a nice picture there showing a wildfire by Brent Stockwell's group, who coined the term ferroptosis. So what happens is that an oxidized lipid radical can then go on and oxidize another neighboring lipid. So once you start it, presumably you can't stop it. And so this is really exciting for those of us dealing with cancer cell resistance, acquired or intrinsic resistance. So the major factors that drive sensitivity to ferroptosis are the presence of polyunsaturated fatty acid containing phospholipids. So that's a mouthful, but those are lipids that can be oxidized through ferroptosis. And of course, the presence of iron, redox-active iron, that can oxidize these lipids. And also the presence of the antioxidant defense of the cells, so where the cells can protect themselves against lipid peroxidation. And why ferroptosis is so exciting for kidney cancer is the fact that the HIFs, primarily HIF2 alpha, drive the accumulation of PUFAs that, like I said, drive sensitivity to ferroptosis, and also the fact that kidney cancer, like many other solid tumor types, have much more iron than surrounding tissue. So those together provide a selective therapeutic window for the selective targeting of tumor cells versus normal tissue. So when I started this journey, this was actually before the term ferroptosis was coined, and that's why government grants are so fantastic, because they let you follow the science. And when I started, we were really looking for inhibitors of HIF2 alpha. HIF2 alpha was coming to the forefront as a driver. Belzutifan was not yet discovered, nothing was developed yet. And so we did a high throughput screen for inhibitors of HIF2. We found compounds that did it, but the mechanism of action wasn't that clear, the compounds weren't that great, and so no one was very excited about that. But when you do the work, you're excited about it, and so we managed to convince the NIH to fund a phase 1 grant for CUDA Therapeutics, a startup that I founded, to develop these compounds, make them more potent, figure out how they work. And we did come up with a novel compound, KD025, proved the mechanism of action, really, really great activity in vitro, but not a good compound in vivo, not very bioavailable. But that helped us secure DoD funding for SAR, lead optimization, to identify a more bioavailable derivative, KD061. And with that then, so this is a journey, it's a long journey. With that then we took 061, went back to the NCI and got a phase 2 grant to fund all the studies needed to get our drug through to IND. And for those of you who have done this is an expensive thing. $2 million sounds like a lot, but really doesn't get you very far. And so we were sweating in our boots, really trying to raise funds for an unproven mechanism of action, new drug, new person, but we're really excited. In June of this year, we received the DoD PRMRP Therapeutics Development Award for $5.6 million. So they didn't cut it. We were really biting our nails to get our drug through to all the studies needed for IND filing to get our drug to the clinic. So a little note on the molecular mechanism of action. KD061 and related compounds target this novel protein known as iron sulfur cluster assembly 2 that no one has heard of. But what happens is that when you block this protein, it's a non-essential protein involved in iron regulation within the cell. When you block this molecule, cells perceive that they don't have enough iron. Nothing really happens to them, but they just panic and they don't think they have enough iron. They activate what is known as the iron starvation response, and that promotes iron uptake, leading to ferroptosis, like I mentioned. But remember, I started with this HIF2 inhibitor screen. So what happens as well is that because HIF2 regulation and iron sensing are intricately linked, so activation of the iron starvation response also shuts down the translation, the synthesis of HIF2 alpha. So we have a dual effect in inducing ferroptosis and blocking HIF2 alpha. So a bit of data here. These are Western blots showing kidney cancer cells treated with KD061, increasing concentrations to the right. And we can see that HIF2 alpha goes down when treated with KD061, and at higher concentrations also HIF1 alpha. And I won't go into great detail, but these proteins, IRP2 and transferrin receptor, TFRC, are proteins involved in the iron starvation response, and they go up. So we're activating the iron starvation response, shutting down the HIFs. And this is ICP mass spec, so iron content within the cells. Treatment with 061 or 025 significantly increases the iron content of the cells. And here we have a cell viability assay. And what it shows is that the cells in the blue line are treated with an increasing concentration of 061, and cells die like you would expect. If you co-treat them with ZVAT, the apoptosis inhibitor, nothing happens. If you co-treat them with NAC, the antioxidant, you completely protect against cell death. So we confirm a mechanism of iron and oxidant-dependent cell death, i.e. ferroptosis. So this is what we're going for, in vivo activity. So oral dosing of animals once a day, and here we have the RENCA model, so RENCA envelops C. mice syngeneic tumors. On the y-axis, you have tumor volume, x-axis, the treatment day. So the black line shows vehicle-treated mice, so the rapid growth of tumors without any treatment other than the vehicle. And the blue and red line show nice dose-dependent effect of KD061 in blocking tumor growth. PD-1 doesn't work very well, and we can talk about that later. But we have a similar effect in the LVRCC model from Ari Hakimi's group. Again, the black line shows rapid tumor growth when treated with vehicle, and blue and red lines really show exciting anti-tumor efficacy, especially at 50 mg per kg. Again, belzutifan and PD-1 didn't work well, and we can discuss why that is, but we're really excited to have a new molecule, new mechanism of action, and great anti-tumor efficacy as a single agent. So to summarize, this is over a decade of work driven by the science, starting from looking for inhibitors of HIF2 alpha, ending with a novel compound orally available that induces ferroptosis in vivo. And this was funded by NIH and DoD grants. Thank you to the DoD. And although I've anticipated our studies in 2028. We think we can get them in the clinic by 2027. So acknowledgement to the funding bodies. That's my lab, but many other people contributed to the work. So thank you.
Questions?
While we're waiting for the microphone to get here in front, I'd like to just ask you a question. First of all, congratulations. Let me just ask you, how key to the success of this drug do you feel like the lipid-rich environment and the HIF environment is for having the efficacy? In other words, do you think this could potentially be efficacious in other cancers as well that don't have those two components?
Well, it seems to be connected to HIF. We know that HIF inhibition has an anti-angiogenic effect, so that's more of a cytostatic effect, and so we're quite excited and more excited about the ferroptosis effect. And that can be driven by iron, having more lipids, having less antioxidant defense. And so we've seen many other cancer types respond depending on what they have of this. For example, melanoma, because they have lots of ROS, so they respond really well. And so we think this can be a broadly applicable therapy, but kidney cancer, because of the lipids, really provides low-hanging fruit, and that's where we're going first.
Thanks.
Great talk, May. So I was going to follow up on what Dr. Walker mentioned. Is the efficacy in vitro that you showed us unlinked to HIF2? I mean, or linked to both? You should have, I mean, it's ferroptosis inducer and HIF2.
In vitro, absolutely, it's unlinked to HIF2, because we know if we block HIF2, cells do perfectly well in vitro. They don't need HIF in a cell culture setting. They only need HIF when they're growing in 3D with blood vessels and hypoxia.
So if I suggest, there are many tumor types in RCC that have ferroptosis. Dr. Lisa Hensky, obviously from several years now, talking about ferroptosis. Renal medullary carcinoma is another tumor, and we published on this recently, where there is also ferroptosis is a. So I think I would suggest that you also test your KD061 in chromophobe and in RMC, because we really need something for ferroptosis. I think this would be a good.
To comment on that, we have talked about the chromophobe, and the problem with chromophobe is that the cell models aren't great. We have like 2 or 3 cell lines, and they grow really slow. And so what we found is that in culture, in vitro, they grow so slowly that if we treat them with 061, they kind of grow a little more slowly or they die, but it's not a really nice therapeutic window that supports going into an animal. So if we had models that actually grew faster, that we could establish tumors more quickly, then that's something we could try. But we're limited by the models currently for chromophobe.
One final question.
Hi, Jonathan Yingling from Marcus Biosciences. I was curious in your last slide whether the one potential interpretation of that data is that the primary mechanism of action for your molecule actually is perhaps not HIF2 alpha, and that's why belzutifan is not working so well, so not dissimilar from the question that you were just addressing. Just curious what you think about the mechanism of action of your novel compound and how much of it's actually HIF2 alpha dependent versus other mechanisms.
So the HIF2 question and the belzutifan question can be slightly separated, because I'm not entirely clear, maybe people in the audience can address this. So mouse HIF2 alpha is slightly different from human HIF2 alpha. It's got a few mutations, a few amino acid changes right in the region where belzutifan binds. And so I don't know if the reason that belzutifan didn't work is because it's mouse HIF2 or whether HIF2 is not a driver for this tumor type. But yes, if belzutifan did not work and it binds mouse HIF2, then I would agree with you that ferroptosis in this case is the primary driver. But we need to establish that functionality first, which is not our focus, but point taken, thank you.
Great, thank you so much. All right, great. Next we have Dr. Vincent Zhu, who's going to be talking about KIM-1.
Thank you so much, and thanks for this opportunity to share some of the updates on from bench to bedside, advancing KIM-1 as a tool for clinical decision-making in kidney cancer. So my disclosures.
If you look at it,
I really would like to start with the acknowledgements, because I think in some way this is the most important part of the talk. And we just heard from Dr. Rathmel about the importance of community in RCC. And really, I think my own experience, this has been so much the case. And this KIM-1 story has involved, not really me, but really a whole community of people, which started even a decade before I knew what KIM-1 was. This started largely in the laboratory of Dr. Joe Bonventry, who initially characterized KIM-1 in renal injury, followed by many folks. And I would take more than 6 minutes just explaining how other people have helped me, but just to focus on the folks who set me on this path of KIM-1, including Dr. Rupal Bhatt, Dr. David Braun, who's here on the stage, Dr. Tony Shueri, Dr. David McDermott, and many, many others. And in addition, the fellows who work closely with me, including Dr. Clara Steiner, Dr. Mark Macalani, and Dr. Liliana Ascioni, and many other people, most of whom are here in this room. This is a community that includes industry and academic collaborators, and really importantly, our funders, CDMRP, Conquer Cancer Foundation, the Kidney Cancer Spore, as well as an R01, where the PI is Dr. McDermott. And so, as many others have mentioned, the CDMRP has really had a central role in kidney cancer research and enabling these studies. And as part of the Kidney Cancer Academy, where we're led by the Academy Dean, Dr. Brian Reaney, and the Board of Advisors shown here, it's really been integral in not just funding support, but really the theme of community, mentorship, collaboration with other members of the Academy, as well as career development. So I'm very grateful for this. We know from now, really up to 20 years ago, that KIM-1 is overexpressed in kidney tumors, especially those coming from the proximal tubular epithelium, and it's shed into the circulation. And subsequently, it's been shown that with work from Dr. Bonventry and Dr. Bhatt, that among RCC patients, KIM-1 is not just elevated in the blood, but it's elevated in the blood using banked samples up to 5 years before the kidney cancer diagnosis. And this really led to the question of whether we can validate KIM-1 as a potential circulating biomarker to detect kidney cancer, but also to risk stratify kidney cancer and hopefully to indicate likelihood of response or as an early response biomarker in kidney cancer. In this setting, we've looked at KIM-1 in a multitude of trials across the disease spectrum in renal cell carcinoma, including mostly clear cell RCC, but also papillary RCC more and more. And this includes the IRK2 study, several adjuvant trials, and also now several metastatic trials with more coming soon. I just want to touch on the pre-nephrectomy data. We know that among patients who have a renal tumor, those with higher KIM-1 at the time of having a renal tumor are more likely to have aggressive malignancies, are more likely to have recurrence of the tumor after nephrectomy, and ultimately a higher chance of dying from kidney cancer. And this was shown with several collaborations, including with Johns Hopkins, folks at Dana-Farber, the World Health Organization, the Blochen Cancer Institute in Moscow, as well as Würzburg in Germany. We also see now from several large adjuvant trials that persistent KIM-1 after nephrectomy is associated with recurrence. And this was seen in the ASSURE trial, Checkmate 914 Part A and Part B, and recently in investigation led by Dr. Laurence Albiges from iMotion 10, really showing us a strong prognostic adjuvant biomarker. And what's interesting is that this also does move forward into the metastatic setting. In both Checkmate 214 and Javelin Renal 101, as well as other studies, we see that high KIM-1 is negatively prognostic in metastatic RCC. In addition, high KIM-1 is associated with aggressive clinical, genomic, and transcriptomic features compared to patients with low KIM-1 in the metastatic setting. And you're going to hear a lot more about this tomorrow from Dr. Macalani's talk. But some of the most fascinating data has been on the potential predictive value of KIM-1, because in my opinion, this may help us to make treatment decisions, which is really what we're hoping for from a biomarker. And so we have from Dr. Albiges' work, along with the Genentech team from iMotion 10, we know that among patients on iMotion 10, benefit from adjuvant atezolizumab was really limited to patients with high KIM-1. And in fact, if anything, disease-free survival is numerically worse among people getting atezo in the low KIM-1 subgroup. So this is really fascinating and points towards some of the possible immune roles of KIM-1 in RCC. A similar signal was seen in Checkmate 914 Part A with nivolumab. In the metastatic setting, we recently learned from two trials, Checkmate 009 and 214, that the KIM-1 ratio, which is defined as the percentage change in KIM-1 from baseline to 3 weeks, really is a strong predictor of response to immunotherapy regimens. And it was particularly striking in Checkmate 214 that patients with a KIM-1 decrease at 3 weeks, those were really the exceptional responders. And the converse was true. Patients who failed to have a favorable KIM-1 ratio by the 3-week mark after 1 dose of ipi/nivo were generally not exceptional responders to ipi/nivo and had quite poor outcomes. And so these data really point the way, I think, towards the question of can we validate these questions to make clinical decisions? And I'm just going to give a few examples, but I think these are questions that I would really love for input from everyone in this room, including the exceptional experience in clinical trials we have in this audience, to try to move this forward. One question is, can we use low KIM-1 to figure out who benefits from adjuvant immunotherapy and who does not? Pembrolizumab is the currently approved adjuvant immunotherapy for RCC, and even though we know that Pembro has an overall survival benefit in the KEYNOTE-564 population, is this really among all patients, or is it really being driven by patients with high KIM-1? Question number 2 is the converse. For patients who didn't quite meet the adjuvant immunotherapy clinical criteria, are we missing some patients who have high KIM-1 who actually could benefit? Patients, for example, with papillary or translocation RCC, patients with clinical criteria that maybe didn't quite reach the threshold, but based on KIM-1 actually have very high clinical risk. Finally, is it possible that KIM-1 can help us give ipi/nivo better? We give ipi/nivo because we hope for the slam dunk, and we would like to give ipi/nivo to more patients, but one of the main reasons that we're held back is that fear of rapid clinical progression within the first 12 weeks. And many patients who get ipi/nivo who don't respond fail to make it to second-line therapy. And could we do better for those patients? Perhaps by assessing the KIM-1 ratio, we can single out the exceptional responders, and for people who will not have a good response, ahead of time divert them to a more effective regimen. And so with that, I would really like to thank again everyone who helped support me in this journey, and I would welcome any questions.
Amazing presentation, a lot to unpack, probably time for 1 or 2 questions.
That's an excellent question, and we've looked at this quite extensively now. So there was a recent abstract, which was led by Dr. Clara Steiner, where we looked at a highly multiplexed proteomics panel examining multiple kidney injury biomarkers along with KIM-1. So we looked at cystatin, we looked at EGFR, we looked at TNF receptor 1 and 2, all these classical biomarkers of kidney injury. And what we saw in bioinformatic analyses was that, first of all, at a very basic level, KIM-1 is not associated with serum creatinine in the setting of people with kidney cancer. That's not true in patients without kidney cancer. In people without kidney cancer, KIM-1 is associated with benign kidney injury. But the KIM-1 shed from tumor is so large that it overwhelms whatever's coming from benign kidney. What we also see as a correlate to that is that all the other canonical kidney cancer biomarkers form kind of a renal injury module, which is completely internally associated with each other, but not associated at all with circulating KIM-1. So it does appear true that among patients with kidney cancer, adjustment for CKD or GFR is probably not necessary when looking at KIM-1.
Great. Thank you so much. And last but not least, we have Wayne Marasco, who's going to be presenting about redesigning CAR T cells for solid tumors.
Hey, everyone. So I'd like to thank the organizers for inviting me to talk today. And I'd like to follow up in Brian Lewis's and Kim's sort of introduction of why an infectious disease physician, which I am, would be working in the field of kidney cancer. So 22 years, almost to the day, I developed painless hematuria following a run from Waikiki Beach up to Diamond Head Crater and back. And the reason I was there is because I was on my daughter's Make-A-Wish Foundation trip. having completed 2 years of chemotherapy. So there I was, a father of 5 young daughters diagnosed with blindsided, diagnosed with renal cell carcinoma. And when I was in Beth Israel Deaconess Hospital, actually in my hospital bed, I had my lab bring over some papers on CAR T cells, which were the new sort of thing at the time, but not heavily, investigated. And I wrote a grant in my hospital bed on CA9, which seemed to be the molecule du jour at the time, and the thing got funded, and that was the beginning of the 22-year journey to get us to where I am now. There are a number of clinical trials listed here on CAR T cells. The first one, which was conducted by Lemire's, I only bring this by way of review, really used CA9 as the antigen. It was a first generation of co-stimulatory domain, and there was severe hepatic toxicity or secondary to biliary duct expressing CA9. That really impacted the field for decades, thinking that it was not druggable, and thus the study sections were quite difficult to get through. There's been some more recent work in this target shown here by Arsenal Bio. CRISPR has done a clinical trial on anti-CD70, and Allogene has done another clinical trial on CD70. I just put them here by way of review, so let you know we're not the only ones in the field. However, what's different about what we're trying to do is our CAR T cells are dual targeted. The affinity is fine-tuned to decrease, improve safety, and we do immune restoring capabilities at the tumor site to change the tumor microenvironment. I'm not going to go into all this in detail, but let's talk about efficacy. So if you look at the panel on the left there, that is surface expression of CA9 and CD70. The second column shows you CD70. It is not universally expressed, so therefore there are targets that are CD70 negative. The left column on the bottom, you can see, on the left column, you can see that almost all tumors express CA9, so it is still the molecule that is most, desirable. The killing data in the immunofluorescence just shows the specificity of the dual targeted CAR for both targets, and on the right is humanized animal studies that show that the vector, the CAR T cells expressing the CAR, D4 CAR, dual targeted immune restoring CAR, effectively cure the animals from tumors, and in these animals we don't even see the tumors after the therapy. Safety's been the biggest concern here because of what I told you of CA9 being expressed on cholangiocytes. We spent over a decade working this out to be able to develop high avidity, low affinity antibodies as the targeting moiety. And why would you want that? Well, we certainly don't want to target CA9 if it's in limited quantities because a high affinity targeting residue will bind to it. So you want only targeting molecules that are recognized high density. And that's what we engineered. The D-Storm slides in the upper middle show that we created cell lines with densities of CA9, that single molecule resolution on the surface of RCC patient tumors at the same level, so we know we're working in the right physiologic range. G250 was the monoclonal antibody that was used in the Lemire's trial of murine monoclonal in 2004, 2005. And you can see in the middle bottom panel, that's survival. Our antibody, cures the animals of tumors, whereas G250 did not. And on the bottom right is fratricide showing that even though CD70 is expressed on white blood cells, we do not get any evidence of fratricide, meaning the tumor cells are killing other T cells. In terms of immune restoring, this is probably, in my opinion, the most important part of this, changing the tumor microenvironment. So we are really experts at humanized mice. We've made thousands of them at this point. So we do either CD34, humanized mice, stem cell reconstructed, or now working with Jackson Laboratories on PBL models. If we do CAR T cells without delivering a payload to the tumor site, we do not ever cure the animals of tumors. If we deliver monoclonal antibodies at the tumor sites, which is what we're doing, we secrete antibodies locally, checkpoint blockade inhibitors locally at the tumor site, we can get complete cures, and that's shown in the bottom right. This is what, lastly, the payload that we're delivering, we're following Checkmate 214 by delivering a bispecific antibody, anti-PD-1, CTLA-4, so we're mimicking the Nivo and Pembro clinical trials that were conducted. You can see on that left panel the combination of Neo and Pimbo is not as effective as a bispecific antibody against those same targets. These are all our antibodies. We designed them all from scratch. And this is a common theme of bispecific antibodies for those that don't know it in the audience. A cocktail is almost never as good as a bispecific because the anchoring of one really affects the activity of the other. And then the cartoon describing what we're trying to do is on the right bottom there. That is really a dual targeted CAR targeting CA9 and CD70, and at the tumor site delivering an anti-PD-1 CTLA-4 bispecific antibody. This was quite a challenge. These vectors are stuffed for those that work in this field. It's about 11.8 kb. So there were issues and concerns. It would never be able to get to a therapeutic dose. I will tell you, never mind, it's not important. That we have been able to achieve that. So we're now at the point of preclinical studies, if there have been or are in the finishing stages. We have made cGMP lot of the clinical trial vector, ready to go into clinical trials. Yufei Wang in my laboratory and Vincent Su up on the stage here are working on the clinical trial design. We're working with the team at Dana-Farber to be able to do the cGMP manufacturing and file for an IND. So we hope to be able to complete all this in the fall. We're on schedule to be able to complete this and file an IND sometime, hopefully in September, maybe October. And I'd like to thank all those individuals in the very bottom. This is quite of a team effort. Expands the oncologists at Dana-Farber, the pathologists at Beth Israel Deaconess, and other manufacturers, people that are involved in the manufacturing as well. So I'd like to acknowledge Yufei Wang, most importantly. Yufei, stand up. She's led this whole effort. And she is the example of what Kim was talking about today. This is an up-and-coming star, and the program is advancing because of her dedication. So thank you very much.
Time for one or two questions. Okay.
Okay, thank you.
Thank you.
You know, I just want to begin this session by congratulating the speakers. I don't think I've ever heard such a fantastic group of 5-minute talks and staying on time as this group up here on the stage. It was just amazing. Thank you guys, it was incredible. So we're going to go ahead and go into the moderated session, but before that, I really believe in audience participation. So I'm the immediate past chair of the programmatic panel for the Kidney Cancer Research Program of DoD. I've actually alerted the photographer because I'm going to call out some of the amazing initiatives that would never have happened without the DoD support. And if you have participated in these, I'm going to ask you to stand up and remain standing until I get to the end of the list, and then we're going to take a picture, and I think it's going to be impressive. So first of all, I want to mention the Clinical Trials Consortium. This was an amazing activity. It was clearly a brilliant idea. It was immediately supported by the panel, and I think is an incredible success story which would not have happened had it not been for the support of DoD and the KCRP. So any of you who have been participating, this includes members on the stage, any of you who have been participating in that Clinical Trials Consortium, would you please stand up right now and remain standing? Your institution or you, yeah, and it's important that you stay up. Don't let Tony leave, guys. Ban the door back there. Okay. Now, one of the other amazing initiatives of the DoD has been this Academy of Kidney Cancer Researchers, bringing up this next generation. in an academy without walls. Any of you who have been involved in this academy as a mentor or as a mentee, please stand up now. Okay, you're going to stay standing. Yeah, don't be shy. All right, fantastic. Anyone here now who has actually received funding for their research from the DoD program, would you please stand up now? Stay standing. All right. Anyone who has not actually gotten support, but who is given service by serving on one of the study sections or programmatic panels for the DoD, would you also stand up now? Stay standing, please. And finally, I would like any of the advocates who have been involved in acquiring or sustaining funding for this initiative, would you stand up if you're not already standing right now? All right, let's let the photographer get a picture of the impact on this community that this program that has just been defunded has made. All right, fantastic. So one of the things that I would like to ask Brian, I presume you're still in the audience because I spoke to you ahead of time and asked you if you mind. Yeah, there you are. Would somebody give Brian a microphone? I just asked him if he could give us an update on the efforts to try and reverse this devastating loss of funding through the Kidney Cancer Research Program. Brian, are you able to speak?
Yeah,
oh, there you are. Excellent. Well, there's somebody back there that kind of looks like you. Okay, great. Come on.
I'm going to get screamed at because we're running behind
Why don't
schedule.
you come on to the microphone?
I'll be literally 30 seconds.
We're not. We have an hour. We're doing good. Come on.
Yeah. I hear stomachs growling out there.
Okay.
Just real quickly, there's been slight encouraging news. We've been slashed, as everyone knows, to 0. However, the House Appropriations Committee met and did their markup, in the Defense Subcommittee Chair, Ken Calvert from California, Tom Cole from Oklahoma, both said in public on the microphone that when the bill comes over from the Senate, they're going to try to make the CDMRP right again. And in the markup bill, they put 15 million back into the House bill, but it hasn't, they're still at 0 in the Senate. So we're starting to see a little bit of movement, a little bit of the, I guess the Republicans standing up to the administration and saying enough is enough, we're gonna fight back a little bit here. So we're gonna need you this fall and going forward, so.
Thank you. Thanks to everyone who's been trying to make the efforts. Okay. So this is, I guess, where we open up the microphones to the folks now to talk about sort of your thoughts on earmarked funding and the importance for kidney cancer research, and not just through the CDMRP. There's other sources of support as well. For example, AACR is now supporting an annual meeting on kidney cancer that's been very, very impactful. So this is a time for you all to speak up.
I'll start. I mean, I think that, you know, this room and what you've just done is an incredible testament to how, by bringing people together with funding support, you create a community. And the thing about these types of things is they're somewhat autocatalytic, but they do require continued support to allow that autocatalysis to occur. And if you end up having breaks in that and uncertainty, it has a tremendous damping effect, and the intellectual sort of product that's created from this is something that we really have to make sure we foster and continue to develop. And I think it's, you know, it's not only the funding from these various sources you're talking about, but in terms of industry partnership, as we move forward, if we are starting to see some issues here, the question really is, how can industry help us paper over some of these gaps, because this is a resource that we really can't destroy.
Thank you, Eric.
I just like.
Yeah, Wayne, please.
Keep talking. Just keep talking. Yeah.
I'd just like to say that CDMRP was really totally responsible for the funding that got us this far. I mean, this took years of funding to take something from conceptual all the way to a product that is going to be submitted, for an IND. So this is really grassroots stuff that It's the continuous support from this group that has allowed this project to move forward. And I just want to say that obviously I'm a supporter of it, but it's been transformative in terms of getting this kind of work off the ground, and I'm sure other members of the stage would agree with that, and the audience. So I'd just like to
And thank you all for highlighting that in your talks. I thought you did beautiful jobs across the board of doing that. Let's have Kim to talk, and then I guess I realized I wasn't reading my watch correctly. So Kim, please, last comment.
So what I was going to say, one, this is a terrific session, and I think it highlights where the field is, where we got TKIs, we learned how to use them, we had immunotherapy, we figured that out, and then now we're at an inflection point where vaccine, CAR Ts, novel ways that we target the epithelial cells, and great biomarkers. It's going to get really messy and complicated. So I wondered if the panelists have thoughts on how we coordinate all of this to get to sort of our next plateau steady state. How do we coordinate all these efforts?
Well, I think it's a question of, it's interesting, Kim, you say that, because we're having, the KCRC, we're having a programmatic review meeting on Saturday morning, and it's going to be a question then of what are the priorities. And, I think we need to, prioritize some of these novel mechanisms of action, in terms of, we know that TKIs are great and they're not going away. Checkpoint blocking antibodies are great, but in terms of where I think the real sort of intellectual effort needs to be expended is probably on the newer cellular therapies, and coming up with ways to develop those from a therapeutic perspective, I think is going to be one of the major foci, because it's a huge effort that we saw with what Wayne has said. But finding a way to actually refine this in a way that we can do it more efficiently is going to be critical.
Okay, well, let's keep the conversation going during lunch. We're ending now the morning session. I want to thank both the speakers here as well as the ones earlier in the day. It's been a fantastic morning. Thank you all very much.